The overall goal of this procedure is to determine the particular alterations in DNA repair protein expression or expression of proteins related to apoptosis that are frequent in areas surrounding colon cancers and thus likely to be central to progression to colon cancer. This is accomplished by first collecting tissue samples in formalin jars either from colonic mucosa from normal colonic mucosa during colonoscopies as control tissues or colonic mucosa adjacent to colon cancers from colon resections as experimental tissues. After formalin immersion for 4 hours, these tissues are transferred to 70% alcohol overnight, followed by embedding in paraffin blocks. The second step of the procedure is to cut tissue sections from the blocks and transfer them onto microscope slides. The third step of the procedure is to immunostain the tissue sections for possible proteins of interest using a sequenza rack. The final step of the procedure is to view the tissue sections in a photomicroscope. Ultimately, results can be obtained that show high levels of expression of one or more proteins in colonic mucosa that has no local neoplasia, and reduced expression of these proteins in field defects surrounding colon cancers, indicating the importance of their reduction in progression to colon cancer. For example, this image shows brown staining indicating high levels of expression of ERCC1 in colonic mucosa without colonic neoplasia. This next image shows brown staining indicating high level of expression of Pms2 in colonic mucosa of a patient without colonic neoplasia, while an area of colonic mucosa near a colon cancer shows one crypt with high Pms2 expression and some with low expression, and another area near a colon cancer with absent expression of Pms2.
Hi, I'm Hoy Nguyen from the laboratory of Carol Bernstein in the Department of Cell Biology and Anatomy in the College of Medicine at the University of Arizona.
I'm Christy Lustino.
I'm Alex Fasista.
I'm Nadia Hasuna, and we're all from the Bernstein Lab.
Today we will show you tissue sections taken during a colonoscopy and a procedure for cutting sections from these tissue samples.
We will then demonstrate the procedures for immunostaining these tissue sections.
We will also show you how we examine our tissue sections with our microscope.
The tissue sections are examined for the expression of proteins, which may be relevant to the progression to colon cancer.
We use these procedures to find that deficiencies in 2 DNA repair proteins, ERCC1 and Pms2, appear to be among the first steps in initiation to a field defect that can lead to a colon cancer.
We also use these procedures to eliminate other proteins of likely importance in early steps in progression to colon cancer, such as DNA repair protein, Ku86, and apoptosis-related protein, CcOI.
So let's get started.
Biopsies of normal colonic mucosal tissues are obtained from patients during a screening colonoscopy and placed in jars of formalin. After 4 hours, the formalin is replaced with 70% alcohol. Similar samples of tissue are obtained from areas of colon resections, 1 centimeter and 10 centimeters away from colon cancers or adenomas with advanced neoplasia. The tissue samples are sent to a histology laboratory for processing into paraffin blocks. Prior to sectioning, the paraffin blocks are chilled in a refrigerator to make them easier to cut by a microtome. Cut tissue sections of 4 microns in thickness from the tissue blocks and float them on water. For comparing the expression of 2 proteins, for instance ERCC1 and Pms2, pick up alternate tissue sections onto 2 slides, until there are 3 tissue sections on each of 2 slides. For example, sections 1, 3, and 5 can be placed on one slide labeled ERCC1, and sections 2, 4, and 6 can be placed on one slide labeled Pms2. The procedure for immunostaining the previously prepared tissue sections should be carried out under a chemical hood with negative air pressure so that the experimenter is not exposed to the vapors. The protocol we use is a fairly standard one. However, to get results similar to ours when you use this protocol in your lab, adjustments may be needed to take into account variables such as water quality, microwave power, and concentration of antibodies. In this video, we will demonstrate Pms2 immunohistochemistry. First, deparaffinize the slides by placing them in 3 changes of xylene for 3 minutes each. Followed by 3 changes of 2 minutes each in 100% ethanol. followed by 2 changes of 2 minutes each in 95% ethanol. Finally, wash the slides in 2 changes of distilled water. Next, place the slides in a buffer solution and bring to a boil in a microwave oven. This is followed by 10 minutes on the medium setting. This step, which is called heat-induced epitope retrieval, breaks the crosslinks formed in the tissue sample during formalin fixation and enables the antibody to recognize the protein of interest. After cooling on ice for 20 minutes, the slides are washed in PBS and distilled water for 3 minutes each. The slides are then placed in a solution of 3% hydrogen peroxide for 20 minutes. The purpose of this step is to block endogenous peroxidase activity in the tissue. Furthermore, by blocking this activity, we reduce the nonspecific background staining that may be caused by the antibody. After 20 minutes in the endogenous peroxidase block, the slides are washed in distilled water and then PBS for 3 minutes each. For immunostaining, place the slides into flat, narrow staining racks called sequenza and rinse with PBS. The slides being stained for Pms2 or ERCC1 are then exposed for 10 minutes to an added treatment of 3 drops of a proprietary solution called Sniper, which acts to reduce non-specific staining of background proteins. After rinsing with TBST buffer, the slides are incubated with the antibody to Pms2. After 2 hours, rinse the slides 3 times with buffer and then add 100 microliters of biotinylated secondary antibody at a 1:100 dilution. Incubate for 30 minutes at room temperature. After 30 minutes, rinse the slides 3 times with TBST buffer. Add 3 drops of Vectistain ABC reagent to each slide and incubate at room temperature for another 30 minutes. After the incubation, wash once with TBST and transfer the slides from the sequenza into a slide rack in PBS. The slides are later immersed in DAB substrate for up to 5 minutes with watching to ensure that the brown staining does not get too dark. This is followed by counterstaining with a dilute solution of hematoxylin for 10 seconds. Rinse slides thoroughly in tap water, followed by deionized water. Dehydrate the slides 2 times for 2 minutes each in 95% ethanol, 2 times for 2 minutes each in 100% ethanol, and 2 times for 2 minutes each in xylene. Finally, add a couple drops of mounting medium to each slide and apply a cover slip. Now, Hoi Nguyen will examine a stained tissue section under a modic photomicroscope for expression of ERCC1 in crypts of the colonic mucosa.
We will look first at a section of colon tissue under a Motic photomicroscope with a 20x objective lens. The yellow arrow is pointing to a colon crypt. There are roughly 200 colon crypt cells in this crypt. These are the stroma cells. This is the crypt lumen, and this is the colon lumen, in which feces passes through. There are 3 different types of colon crypt cells. Goblet cells are shaped somewhat like a wine glass, and their nuclei are at the outer edge of the crypt, shown here and highlighted in red. They also have a ballooning out section filled with white mucin granules in the part of the cell on the inside of the crypt, shown here. The other 2 types of cells look somewhat alike in our stained sections. They are the enterocytes and enteroendocrine cells, and their nuclei are also at the outer edge of the crypt, at the base of the cells, indicated here by the red circles. This particular section was stained for ERCC1. The brown stain shows the location of ERCC1. Almost all of the nuclei in all crypts in this section show normal high level of expression of ERCC1. The blue stain is hematoxylin, which stains DNA in nuclei, as shown here. The nuclei in the crypt have more brown than blue because of the high expression of ERCC1. Now we will navigate through the tissue with the 10x objective lens. Notice how most of the colon crypts are showing high levels of ERCC1 expression. Here we have an area with a few crypts showing lower ERCC1 expression, indicated by the blue staining nuclei. As we approach the end of the tissue, we notice another area with lower ERCC1 expression. We will now go back and look more closely at the crypts that were deficient for ERCC1. Looking with the 20x objective lens, we can see the lower ERCC1 expressing crypt next to a higher ERCC1 expressing crypt. Notice how the ERCC1 protein is more heavily expressed in the cytoplasm of the deficient crypt, indicating that the ERCC1 is produced but unable to get into the nuclei. Now with the 40x objective lens, we can see more clearly that the ERCC1 was unable to get into the nuclei of this deficient crypt. Compared to the highly expressing nuclei of the neighboring crypt, with little ERCC1 in its cytoplasm. This tissue was taken as a biopsy from a patient with a previous colon cancer.
Next, Nadia Hasuna will examine a stained tissue section for expression of CcOI in crypts of colonic mucosa.
We can go through a tissue section immunostained for cytochrome C oxidase 1, or CcOI, using a 40x objective. CcOI is a cytoplasmic protein and is not expressed in the nuclei, which is why the nuclei have stained only blue. CcOI is expressed in the cytoplasm, shown with the brown staining. In this field of view, we can see two colon crypts, one having positive expression of CcOI and one having a deficiency in CcOI expression. Panning through this tissue section, you will notice that the majority of the colon crypts have a positive expression of CcOI.
Now, Hoi Quinn will show CcOI immunostained crypts indicating the stem cell region of the crypt and its function.
We are now looking at a section of tissue stained for CcOI. Reports indicate there are less than 5 stem cells at the base of the crypt, giving rise to the 2,000 total cells of the crypt. The stem cell region is shown in the red box, indicated here. A stem cell with a mutation or an epimutation can take over the whole stem cell niche, or half of the stem cell niche. Here we have crypts that are showing high normal expression of CcOI, indicating stem cells without a mutation for CcOI. Here we have an example of how a mutation has resulted in altered staining of a whole crypt stain for CcOI. Notice the lack of CcOI expression throughout the entire crypt. Especially when compared to a normal staining crypt. Here we have an example of how a mutation has resulted in altered staining in half of a crypt. Notice how this section of the crypt shows low levels of CcOI expression when compared to this section of the crypt, which shows high level of CcOI expression.
Now Nadia Husuna will show CcOI immunostained crypts in cross-section and indicating that some crypts have cross-sections arising from mutant stem cells lacking CcOI.
Here is a tissue section immunostained for CcOI showing colon crypts cut crosswise. We will observe this section using a 20x objective. There are two pairs of fissioning crypts showing positive expression of CcOI, and one fissioning structure showing CcOI deficiency in one crypt. This crypt shows deficiency of CcOI expression in the whole structure, where all of the crypt stem cells became CcOI deficient cells. These two crypts show half conversions, where approximately 50% of the stem cells were deficient in CcOI expression. These stem cells gave rise to the deficient half.
Now Alex Fascista will examine a tissue section immunostained for CcOI and identify elements of the tissue.
For the purpose of identifying some other structures in the tissue, we will observe a section stained for CcOI with a 10x objective lens. Indicated by the red box is a lymphoid nodule, the luminal edge, a crypt, interstitial cells, also called stroma cells, and the muscularis, which extends down the tissue.
Now we will look at a slide showing tissue obtained from the normal appearing area of a resection when a patient had a segment of his colon removed. This area, very near a colon cancer, can be considered to have a field defect from which the cancer arose. Here, some crypts have higher levels and some have lower levels of expression for ERCC1. Christy Lustenau will show us how this tissue is evaluated.
Here is a tiled image taken with a 10x objective lens of a tissue section closely adjacent to a cancer, and it is stained for ERCC1. Next to a cancer, many of the crypts are frequently deficient for ERCC1. We will start to evaluate the level of ERCC1 expression using a 40x objective lens, and the crypts are given a score from 0 to 4 depending on the expression level of ERCC1. Each tissue section on the low resolution image is then dotted with a color: blue for high expression level 4, green for fairly high expression level 3, yellow for reduced expression level 2, orange for very low expression level 1, and 0 for absent expression level 0. Here is the first crypt we will be scoring. As you can see, the nuclei along this edge are almost completely deficient. On the other edge, there is a small amount of expression, and we label this crypt level 1 for very low staining and dot it orange. This crypt has a fairly high amount of staining in almost all of its nuclei, getting a green dot for level 3. By comparison, all of the nuclei at the lumen have very high level 4 staining. This crypt has low expression of ERCC1 along this edge, so we dot it yellow for level 2.
This crypt exhibits both high expression on one edge and fairly high expression on this edge. And we place a green dot for overall level 3 expression. This crypt has reduced expression of ERCC1 and is marked level 2 with a yellow dot. This crypt has high level 4 expression and we give it a blue dot. Overall, this crypt appears to have reduced expression level 2, getting a yellow dot. We consider crypts level 0, 1, and 2, red, orange, and yellow, to have reduced or absent expression, whereas level 3 and 4, green and blue, are considered to have relatively high expression. This tissue section had 44 crypts with low expression and 23 crypts with relatively high expression. Thus, 67% of crypts in this tissue section near a cancer had reduced or absent expression of ERCC1.
Tissue sections cut one after the other from a tissue block, with alternate cuts placed on separate slides, allow us to look at the same colonic crypts on 2 slides. These tissue sections were obtained from tissue very near a colon cancer. We stained one slide with antibody to ERCC1 and the other slide with antibody to Pms2. These tissue sections are shown by Christy Lustenau.
Here we have one of 2 sequential cut tissue sections near a colon cancer, immunostained for ERCC1. On a 10x objective lens, you can notice ERCC1 is highly stained near the lumen, although most of the crypts are not stained. You can also notice the same brown staining in the interstitial cells. At a higher magnification, we see this crypt has high expression of ERCC1. Here we have a second sequential cut tissue section near a colon cancer, immunostained for Pms2. As we move through this tissue section, the crypt, which has stained for ERCC1, has also stained for Pms2. At a higher magnification, we see high expression of Pms2 in the same crypt. Here both crypts are paired. You can notice the high expression of ERCC1 here. Similarly, the same crypt, stained for Pms2, also has high expression. These neighboring crypts do not exhibit high expression, shown here and here. Adjacent crypts, stained for Pms2, also do not show high expression here and here.
We also use immunohistochemistry to determine the frequency of crypts deficient for Ku86 and CcOI. On this slide, Hoi Nguyen will go through an entire tissue section obtained near a colon cancer and immunostained for Ku86. We see that a few crypts are deficient for Ku86 in this cancer-associated tissue section.
We also use immunohistochemistry to determine the frequency of crypts deficient for Ku86 and CcOI near a colon cancer. On this slide, we go through part of a tissue section immunostained for Ku86 using a 10x objective lens. As we navigate through the tissue, we see that most crypts show high level of expression of Ku86. However, as will be indicated by the red boxes, there are a few crypts that are deficient in Ku86.
On this slide, Alex Fasista will go through an entire tissue section obtained near a colon cancer and immunostained for CcOI.
We are now looking at a tissue section taken 1 centimeter away from a colon cancer, immunostained for CcOI. With a 10x objective lens, we can observe a crypt with high expression of CcOI, as well as a cluster of crypts with low levels of expression of CcOI. Here is a fissioning crypt. This portion has high level of expression, whereas this portion has low level of expression. As I pan through this tissue section, we can notice that most of the crypts have a high level of expression. I have previously tallied all of these crypts. There's a total of 189 crypts, and 45 of them are deficient in CcOI. That gives a frequency of about 23.8% deficient. In this crypt, about half of the crypt is highly expressing in CcOI, whereas half of the crypt is deficient in CcOI. We note that CcOI deficient crypts are not as frequent near colon cancers as Pms2 or ERCC1 deficient crypts. CcOI deficient crypts tend to have a frequency of 10 to 20% deficient, whereas ERCC1 and Pms2 tend to be around 70 to 90% deficient near a colon cancer.
We've just shown you how to cut and immunostain tissue sections from colonic mucosa for proteins that may be important in progression to colon cancer. We've also shown you how to evaluate these tissues for deficiencies in certain proteins that are in the crypts.
When doing this procedure, it's important to have 3 tissue sections per slide. Bubbles can occur when applying the media carrying the antibody, preventing crypts or portions of crypts from staining adequately. Just because a bubble prevents the antibody from reacting with the tissue section.
Crypts are usually 60 microns in diameter, and tissue sections are 4 microns thick. So there could be as many as 15 tissue sections through the same crypt. It is usually possible to find the same crypt using the morphology of the crypt as a guide on multiple tissue sections placed on a slide.
So that's it. Thanks for watching and good luck with your experiments.